Welcome back to botany class. Let's move to the next chapter, fifth chapter, plant tissue culture. The topic of fifth chapter is plant tissue culture. Let's look a glance of learning objectives. The learner will be able to perceive the concept of plant tissue culture, cognize the steps of techniques and types, understand the protoplast culture in detail, elicit the list of secondary metabolites, learn plant regeneration and acquire knowledge of patenting, biosafety and bioethics related to micropropagation. These all topics you are going to learn under this heading plant tissue culture. So, at first the concept of tissue culture. There are different concepts, one by one we are going to learn. Then steps in the plant tissue culture, techniques and types. Protoplast culture. What is protoplast? How it is isolated and how it is cultured. And the list of secondary metabolites. There are number of metabolites produced by the plants. These secondary metabolites inhibit the growth of the plant. This elicit the list of secondary metabolites and regeneration, plant regeneration. Then acquire knowledge about patenting, biosafety and bioethics. Let's start from the beginning. What is meant by plant tissue culture, or what's the definition of plant tissue culture? Plant tissue culture is a technique by which the protoplast or a tissue or organs are cultured in a aseptic condition. That means it's away from the normal or natural environmental conditions. Normally the plants grow, there is a normal process, there is a method of reproduction. From the first chapter you learned it very detailed, but here it's the another way, the plant tissue culture technique. A single tissue has the ability to produce the entire plant because of the totipotent nature of the cell. Here plant tissue culture, the plant is cultured, or a tissue, the plant, from the plant there are number of parts, different parts of the plant, a tissue or a single cell, that's called explant. Explant is the part of the plant. So this may be a single cell or a tissue or it's a organ, is cultured to develop or multiply it. A single cell can be multiplied in thousands of plants by aseptic condition. That is, it's away from the normal or natural environmental condition, is called as plant tissue culture. That's the definition. The plant tissue culture is called as in vitro. In vitro. That's a Latin word that means a glass, in the glass or in the test tube. The plant cell can be cultured in a single cell, single vessel, that is a glass or a test tube, a glassware. That is called in vitro culture under aseptic condition, is called plant tissue culture. Then what is the applications of this plant tissue culture? So today you are going to learn the basic concepts of plant tissue culture and the applications. What are the applications of plant tissue culture and the techniques involved in it, techniques, the history, types, techniques, applications and ethics you are going to learn today. So first write about what is meant by plant tissue culture. A single cell has the ability to develop into an entire organism under aseptic condition, is called as plant tissue culture. So this word is important. Explant, the part of the plant. It may be any part of the plant, either root, stem or leaves. So this tissue or cell isolated from the plant and it is able to produce the entire organism is called as plant tissue culture. What are the applications wherever this plant tissue culture is applied? So this plant tissue culture is applicable in various researches, plant researches, mainly in commercial value. It gives commercially important, economical value is important, commercial production of this plant tissue culture, the tissue culture plants within a short period. What's the importance of this? Within a short period, within a short space, a single explant develop into thousands of organisms. So commercially it is very important. Also this is used for plant research, plant researches, and it's used for virus free germplasm, virus free plants. Virus free plants can be produced by this technique, plant tissue culture. The germplasm culture, germplasm is the single living cell. From a living cell, single cell, it produced as an entire organism. Here protoplast, the protoplast, plant tissue culture, protoplast culture. Here protoplast of the plant, if you take the cell, plant cell which contains the cell wall. In this, inside there is a nucleus and all kinds of cell organelles are present in it. So this removing this cell wall and all the things present inside, inner to this is called as protoplast. So isolate this and culture it under aseptic condition is called as this technique, plant tissue culture. So in this method we can create virus free plants, virus resistant varieties, and germplasm, the living part, this protoplast can be selected and this is able to make it into, transfer it into a nutrient medium and make it into an entire plant, germplasm culture. And these techniques are involved to produce the endangered species. There are number of species are risk to extinct. So when the plants, there are rare species, they are going to extinct. That species are called as endangered plants. It's a risk of extinct. So these plants can be isolate, the protoplast of this plant or the cell, a single cell or a tissues can be isolate from a plant and it is transfer it into a nutrient medium and develop it into the entire organism. So these all applications are very important, plays very important role in biotechnology. So under biotechnology, this plant tissue culture, nowadays this plant tissue culture techniques are playing very important role. Then who was the father of plant tissue culture? Gottlieb Haberland in the year 1902. He was the first person who cultured the mesophyll tissues of this plant, Lamium purpureum. One word question, Lamium purpureum. Haberland, at first he was the first person who isolate a cell from the Lamium purpureum plant, mesophyll. From the mesophyll of Lamium purpureum, isolate the tissue and it is cultured in aseptic condition to develop the entire plant. So he is regarded as the father of plant tissue culture. Who was the father of plant tissue culture? Gottlieb Haberland. Haberland was the father of plant tissue culture. So this plant tissue culture contains the basic concepts. So first question, your first 5 mark question you are going to learn today is basic concepts of plant tissue culture. What are the basic concepts? Basic concepts of plant tissue culture includes totipotency. The first concept is totipotency, differentiation, de-differentiation, and re-differentiation. Basic concept. The basic concepts of plant tissue culture is totipotency, differentiation, de-differentiation, and re-differentiation. So, what's meant by totipotency? So, first, this all 2 mark question. What's the definition? Totipotency. Totipotency is the ability of a single cell or the explant. The explant is isolated from the plant tissue, plant part, any part of the plant. So, this explant, it is selected part of a plant. It may be a cell or tissue or organ. So, this explant has the ability, or it has the inherent potential, genetic potential, to develop into an entire organism is called totipotency. So, what is the definition of totipotency? The explant of a plant has the potential or ability, that means the living part of the plant has the ability to develop into an entire plant is called as totipotency. Because of this nature of plant, this tissue culture technique is involved. So, this tissue culture technique is based on this concept totipotency. Totipotency is the ability of the living cell to produce entire organism. Then differentiation. What's meant by differentiation? Now, look at this diagram. Here there is a tissue explant which is selected from this plant part. This from carrot, explant is isolated. So, what is explant? A tissue taken from a selected plant. This is the tissue, selected tissue from the plant. From this selected plant, a tissue is selected. Here this is the tissue, selected tissue, which is known as explant. The name of the tissue is explant. So, this explant has the ability, only a single tissue, single cell is in the form of tissue, few cells or a single cell. It has the potential, inherent potential to develop the entire organism, entire plant. So, single explant has the ability, or it has to multiply into thousands of plantlets, and from this plantlet it able to produce the entire plant and gives yield. So, such process is called as this plant tissue culture. So, totipotency is because of the inherent genetic potential of this explant. Now, differentiation. This plant cell under normal condition, now it is completely artificial, it is not in the natural environment. So, this explant need artificial medium, culture medium, and artificial light, artificial everything, artificial aseptic condition. All the natural conditions are given in the form of artificial to develop into the entire organism. So, this explant is transferred it into the nutrient medium. Here is the vessel or glassware which contains the nutrient medium. The cell has to transfer it into the nutrient medium. You know that when the plant grows, it needs number of minerals, macromolecules, micromolecules, and so on. So, this all macromolecules, micromolecules, and the minerals, everything has to provide hormones, growth hormone, everything can be produced artificially. So, culture medium is very important. So this plant is transferred it to the culture medium. So differentiation, what's meant by differentiation is the process of biochemical and structural changes. So this X plant undergo biochemical and structural changes by which the cells become specialized in the form and function. So this cell able to function. So specialized function of this because of the changes. So this X plant undergo number of changes when it transfer it into the nutrient medium. There is a biochemical changes, structural and functional changes of this X plant is called differentiation. And next concept is de-differentiation. So de-differentiation, before that re-differentiation. Now from the differentiated cell, so here re-differentiation, first totipotency, differentiation and re-differentiation. So what's meant by re-differentiation? From this differentiated cell, at first isolation of X plant, the first process is isolation of X plant from a selected plant. So this tissue is transferred into the nutrient medium. So this nutrient medium, there is a differentiation. So now the cell or tissue is able to structurally and functionally biochemical changes are taking place. That is differentiation. Now this differentiated cell undergo re-differentiation. So this differentiated cells, it forms from here when it is under nutrient medium. This single cell, it able to produce a mass of tissue, that is callus. So it produces a callus. Now the term is callus. What's callus? Callus is the undifferentiated mass of cell. So mass of cells are developed within the nutrient medium. So this differentiation, the already differentiated cell re-differentiated, further division, cell divisions takes place and it produces, the growth takes place and it differentiated again and again and it forms a mass of tissue called callus, is called re-differentiation. So callus formation is very important in this. Then de-differentiation. De-differentiation is the reverse process. Now from the single cell, callus is developed. Callus is the meristematic tissue. So now this meristematic tissue undergo number of changes. So re-differentiate, here it is de-differentiation. So this callus, at first in de-differentiation, callus becomes embryo, early embryo stage, then late embryo stage, and finally it produces a plantlet. During de-differentiation, there is a reverse process of this mature plant cell. Again this mature plant cell produces callus. This is de-differentiation. D is the reverse process. De-differentiation is the reverse process. Re-differentiation is from the differentiated cell, callus formation, embryo, early embryo, late embryo and the production of plantlet. Hope that you understand clear. Once again, this basic concept is very important. Totipotency is the inherent potential of the living cell, has the ability to develop into an entire plant. Differentiation, there is a biochemical changes taking place within this X plant when it is transferred to nutrient medium. When this is transferred to the nutrient medium, there are number of biochemical changes takes place and the cell becomes structurally able to function, and further division of this tissue undergo the formation of callus. So this callus is a mass of undifferentiated cell. So this cell again forms the embryo because it can be developed into the entire plant. So formation of embryo, then early embryo, late embryo, the shape changes, lot of changes taking place. Finally, it produces the plantlet. So number of plantlets from a single cell, thousands of plantlets can be developed. So this process is re-differentiation. Now de-differentiation is the reverse action of mature plant cell to become callus. From callus to mature plant is re-differentiation. From mature plant to formation of callus is this process is reverse action of mature plant to callus formation is de-differentiation, and from this callus to entire plant, formation of callus to entire plant is called as re-differentiation. So don't confuse these two, re-differentiation, de-differentiation and differentiation. These are the basic concepts. So first question is basic concepts of plant tissue culture and protoplast culture. So next question is plant tissue culture. This plant tissue culture, which is called as PTC, plant tissue culture. So what's meant by plant tissue culture? The protoplast can be isolated from the cell. From the cell, the protoplast is isolated and it is culture it into the nutrient medium, transfer of this explant. So explant is very important in this process. This explant is very important. Explant. The explant is transferred to culture medium. So for making this plant tissue culture, making a tissue to entire organism, make it as an entire organism, there are three important techniques are used, three principles. First one is concept, that is totipotency, differentiation, de-differentiation and re-differentiation. Re-differentiation, de-differentiation. Now what are the basic principles? So at first, the first principle is isolation of explant. Isolation of explant. This explant is isolated. Here the explant is isolated from this tissue, this plant part. From different parts of the plant, the explant can be isolated. So first, the explant can be isolated. Then second principle is this explant undergo physical and chemical environmental conditions. So physical and chemical changes allow this explant undergo physical and chemical changes, because this explant is culture in artificial synthetic medium. Artificially we are going to give all the nutrients. So chemically the nutrient medium, this nutrients, minerals, vitamins, plant growth hormones, at different stages the plant provide, we must provide the nutrient alternatively, the different stages when it occurs. So chemical changes, chemically it can be provided. Explant, the treatment of explant with chemical as well as physical. Physical, the environmental condition, because it is grown in a controlled room, controlled condition, within a room or the laboratory. This plant is able to develop. So within the room, naturally the plants are developed in the sunlight, direct sunlight. Sunlight is needed, so it is photo period, day length, day time and the night time, photo period. There is a particular time. This photo period is necessity. And the physical environment includes this light and dark period. When the plants grow, there is a different types, depend upon the plant species, the length of the day and the night may be changed. So according to the species, it can be provided. So explant is treated with physically and chemically. The physical treatment, like environmental condition, the humidity, pH, everything can be given, light, air, everything. And chemically, the nutrient medium has been taken. And the third principle is the explant must be selected or isolated, selected from a isolated tissue. What is explant? The definition of explant is tissue taken from the selected plant can be transferred into the nutrient medium. Transfer of explant to nutrient medium. Nutrient medium. So these are the three basic principles for plant tissue culture, PTC, plant tissue culture. First question is concept of plant tissue culture. Second question is basic principles. These three principles are important. Isolation is very important. Isolation of explant is important, and the explant undergo physical and chemical treatment. That means artificial environmental condition as well as artificial chemical substances, nutrient medium can be provided to allow this explant develop into an entire organism. Third one, this explant can be transferred into the nutrient medium. First, preparation of nutrient medium, prepare all these conditions, chemical, pH, temperature, ventilation, everything can be aseptic condition, under controlled condition. All this environmental, chemical, sterilization conditions can be given, and finally the transfer of this explant to the nutrient medium allow it to grow, to develop the entire plant. So for making this, from this explant, this is the explant, and there are number of changes taking place within a short duration, within a short space. So normally the plant, the tree or a plant can grow in a spacious area in the ground, normal direct exposure of direct sunlight, but it is grown in the laboratory itself within a small glassware, either it's in the form of a small test tube or conical flask. So small glassware is selected, it's enough. The limited space is short space as well as the short duration. Within a short duration the entire process can be taking place. Now what are the facilities given for this process, plant tissue culture? What are the facilities? In the lab or in the room can be sterilized. The room is sterilized room. Number of glass wares are taken. There are number of glass wares. So you know, in the labs we are using normally we are using test tube, petri dishes, conical flask, likewise different varieties of glasswares are available in the laboratories. But this test tubes and the tissue, when we are transferring this tissue from explant, when it is transferred to nutrient medium, we cannot take it in the hand because easily it get affect the cell. The cell we are isolated from the plant. The cell easily get spoiled. Even if you expose it to the air, it gets spoiled. There are number of microbes. So the room must be sterilized condition, that is aseptic condition. So at first, what are the facilities given? These all glassware washing facility. Washing facility in the room. It may be in the laboratory or in a single room, what are the facilities given? The question is, what are the facilities given to the plant tissue culture room? That is first facility, washing facility. These all glasswares or this glass vessels can be washed, and this plant part can be washed. So water facilities, and after washing oven is needed to dry up these vessels. So water facilities and oven for drying these glasswares. So first facilities, washing facilities. Wash all these glasswares. After washing, the oven is needed to dry up these all glasswares. Then second facility is nutrient medium. Nutrient medium is very important. medium. So this nutrient medium can be preparation of nutrient medium. So for preparing this nutrient medium, the nutrient medium also sterilized. The area, the room can be sterilized, the glass vase can be sterilized, and the nutrient medium also sterilized. For sterilizing this nutrient medium, there are autoclave is needed to sterilize this nutrient medium to kill the microbes. There are number of microbes present. It's possible to enter into the nutrient medium. So before transferring this explant into the nutrient medium, it can be sterilized with autoclave. That means high temperature can be allowed to kill the microbes which is present in the nutrient medium, autoclave, and maintain the pH. So pH meter is needed, pH meter, and balance, electronic balance. So these all are to sterilizing the nutrient medium. So it's taken, this facilities must needed, and this all process, when they are transferring this, there are number of equipments are need, forceps, knife. If it is taken as a leaf or in the plant pot, here it is a carrot plant pot. So cutting this, they are using knife as well as forceps. So everything can be sterilized with flamed and cooled forceps. So for flaming facility and cooled forceps, knife, everything is needed. And very important, laminar airflow bench, or laminar airflow chamber. So this all techniques are in front of the laminar airflow chamber. Laminar airflow chamber is the moving of air. It's allowed to moving the air in the same direction. So there is no cross of air, because this explant very easily it gets spoiled. These are single cells and the tissues. If it is in the open air, it gets spoiled. And filter, the room must contain the filter the unwanted air as well as the microbes. So filter, laminar airflow chamber and filter. So this laminar airflow chamber which moves the air in a same direction. So in this laminar airflow chamber, these all procedures can be done. So you may see a number of labs, tissue culture labs. So it is highly aseptic condition. It's a closed room with all given facilities. These all facilities can be provided to develop this explant into entire organism. And this explant must provide to develop into the entire plant the temperature. So incubation period is important. There are 22 to 20 degree Celsius in the inoculation. So inoculant, the temperature can be maintained, and the pH 60%, pH the humidity is the atmospheric air humidity, 60% humidity, and intensity of light, there are 2400 lux illumination is needed, and the photo period 8 to 16 hours photo period. So temperature can be provided, because when the plant cell it undergo this all differentiation, pre-differentiation and de-differentiation, it takes temperature. There is a particular temperature needed, and intensity of light. Artificially we are providing the light intensity, that is 2400 lux, and the photo period, the daytime and the night time. The natural plants which is daytime fully it is exposed to direct sunlight. Here we are providing artificially, so this much light. Photo period 8 to 16 hours, and the humidity. Humidity is the atmospheric air. So 60% humidity also provide to develop into the entire species. So this question is, what are the facilities given to the X plant develop into the entire plant? First facility is washing facility. Washing facility with oven to dry up these vessels, and a nutrient medium. Nutrient medium can be sterilized by using autoclave or pH meter, balance. So these all facilities can be given. Laminar airflow chamber or laminar airflow bench. Then filter the unwanted air as well as microbes from the air. From the air also lot of microbes can be affected, because this X plant or the cell or tissue which is chosen for developing into the entire organism easily gets spoils by microbes. So these facilities can be provided, and this all photo period, humidity and intensity of light and temperature, give a particular temperature, can be provided to grow as the entire plant. So then techniques. So at first, so here today you are going to learn what are the history. Haberland, regarded as the father of tissue culture. So he was the first person. He cultured mesophyll tissue of Lamium purpureum. That's the history of plant tissue culture. And before that, the definition, what's the definition? There is a totipotency, potential of this X plant to develop into the entire organism, and basic concept including totipotency, differentiation, redifferentiation and dedifferentiation. Then this plant tissue culture principles, there are 3 different principles for isolation of X plant. How to isolate the X plant? First, isolation is very important. The plant part, selection of this plant tissue is important. That's called X plant. This X plant has to provide physical and chemical conditions. Physically, environmental condition, and chemically this nutrient medium, preparation of this nutrient medium, and this X plant can be transferred into the nutrient medium to develop into the entire plant. Then finally, what are the facilities given to this X plant to develop into the entire species? And next question is the technique. What's the technique of this plant tissue culture? There are sterilization. First process is, or the process of this culturing plant is sterilization is the main process. At first process is sterilization. So these facilities, already I told you what are the facilities need. These all facilities for sterilizing. Washing is one of the sterilization method, and the nutrient medium, chemicals can be sterilized with the help of this autoclave, and the pH meter, maintaining the pH level of this nutrient medium is important, and this laminar airflow chamber without microbes, because if any microbes enter into the cell, it spoils, it cannot develop into the new plant. So proliferation do not get. So single cell can be multiplied into thousands of plantlets. So this process must be in sterilized condition. And second important process is transfer of this X plant to nutrient medium. So physical sterilization as well as chemical sterilization, sterilization of nutrient medium. Then callus formation. So if this nutrient medium is a sterilized one, and all this intensity of light and the photo period and everything can be given in a proper, maintaining this pH under aseptic condition in laminar airflow chamber and filter all the microbes, it can develop into the callus. So callus formation, next process is callus formation. From callus it develops the embryo. So callus formation, development of callus, inoculation, incubation, and at last hardening. So this plantlet, number of plantlets can be produced. Now this plantlet is transferred to direct environmental condition is the process. So techniques, in the next class you are going to learn in detail about this technique. So sterilization, just you remember what are the techniques involved in this: sterilization, the nutrient medium, sterilization of nutrient medium, and this formation of callus, incubation, inoculation, and this hardening. Final process is hardening. So today you learn first question, the definition of plant tissue culture. Second, that's definition, 2 mark question. Basic concepts of plant tissue culture: totipotency, differentiation, de-differentiation and re-differentiation. Then the principles of plant tissue culture and the facilities given for plant tissue culture. Thank you.